Okay, good afternoon, everybody, and welcome to the first seminar in our fall 2021 seminar series. I want to give credit to Jessie Briggs for setting up this month's seminar series, and she's actually going to be our speaker today. I'll introduce her in a minute. But I also wanted to highlight, we've got 3 other speakers over the next few months. Scott Pfeiffer in October, Peter Narins in November, and Graeme Shannon in December. So please mark these on your calendar. All the Zoom information is in the flyer. And I should say that last semester, we were very focused on underwater presentation. This semester is all terrestrial. So putting people outside or inside their comfort zone, depending on what side of the fence that you fall on. So please mark your calendar for that. I'm going to stop sharing and ask Jessie to bring up her presentation. And as she's doing that, I will introduce Jessie. So Jessie is our first speaker. She's a PhD candidate here in the Department of Biology at UNH in integrative and organismal biology. She has a bachelor's in zoology from Washington State University, and she has a diverse topics that she has worked on over her career. Her bachelor's project was an isotopic analysis of the effects of increased carbon dioxide on the trophic interactions of a desert ecosystem. She's a master's of science in biology from the University of North Carolina at Greensboro, where her thesis was the individual context of ultrasonic vocalizations in wild California mice. And today she's going to be talking to us about a portion of her dissertation research that's in progress, an integrative approach to assessing the impact of noise on Australian black field crickets. So Jessie, I'm going to go on mute and you can take it away. Afterwards, I will monitor the chat for any questions that come in, so you don't have to worry about that.
Okay.
Okay, take it away.
Hey, thank you. Thank you guys all for coming today. So like Jen said, this is the first part of my dissertation. So this is the first chapter, and I'm looking at how different forms of noise influence phonotaxis in female crickets. So as many of us know, noise is prevalent in most ecosystems, including some of the most protected areas in this country. So this is a good map that shows those protected areas. And in most of those locations, background noise levels are almost twice that of the natural ambient levels. And so this is kind of puts more pressure on these animals as far as behavior goes. And so anthropogenic noise is an ecological concern that has been shown to have wide-ranging effects on many animals. And so a lot of the research we've seen has been on the impacts to vertebrates, and we know that there's broad impacts to vertebrates, and some of those impacts include increased predation. We've seen that prairie dogs increase their vigilance in the presence of noise. And kangaroo rats have seen an increase in predation when they can't detect incidental sounds. A decrease in mating opportunities. So for some greater sage grouse, they actually decrease their attendance at leks in the presence of noise from compressors at oil sites. There's also avoidance of noisy areas. Bats tend to avoid hunting at noisy roadside areas. Potential to mass calls, especially with contact calls for animals that rely on those for pair bonding. So we've seen that finches actually increase pair copulations when their contact calls are masked. And signal shifts. I think this is one of the more common studies we see in looking at the impacts of noise on animals, is that in urban areas, birds will actually shift their frequency to better be heard over that urban noise. And then just a few other ones we see is increase in stress, distraction, which has the potential to increase predation if an animal is distracted. And there's also physical damage that can occur to an animal's hearing apparatus. But most of this research has been focused on vertebrates, and not just vertebrates, but vertebrates and airborne noise. Now, invertebrates, though, make up 97% of identified species. And between 1990 and 2013, looking at peer-reviewed publications, they actually only make up 4% of peer-reviewed publications looking at the impacts of noise on animals. So that's a huge disparity between what we see as far as recognized or identified animals go, and then the research done on them. Now, of all those papers, 65% were birds and aquatic mammals. So it's overwhelmingly towards those two taxa. And so insects aren't, or invertebrates aren't the most charismatic animal, you know, so maybe that's why we don't do as much research, but they're actually really, really important for most ecosystems. And, you know, there's a lot of conversation now about pollinator collapse and what that means. So we know a lot about that, but what else do insects and other invertebrates do? Well, one, they're prey species. Like I said, they're pollinators. They're also indicators of environmental change. They help with seed dispersal, decomposition of organic matter, nutrient cycling, and habitat formation and modification. And so insects really are an excellent candidate for studying different types of noise. And they may actually be better models to study sound than vertebrates, even though most research focuses on vertebrates. So vertebrate hearing really has only evolved once, whereas with invertebrates, looking specifically at insects, hearing has evolved 24 times independently in insects. And for some orders, it's actually evolved multiple times independently, okay? And so because of how many times we've seen hearing evolve, we get this diversity of hearing organs. So here is an example of all the areas on an insect's body that we've so far found hearing organs. And we may still find more hearing organs as we continue our research with hearing and insects. Okay, so there's this huge diversity amongst insects as far as hearing organs go, and they have advanced sensory systems, but they also have diversity of organs for sound production as well. And so they often produce sound multiple ways. So some of those examples are percussion, stridulation, tremulation, and timbalation, okay? And so with all that diversity, we can assume that the impacts of anthropogenic noise on insects can also be very complex, because also these invertebrates are under pressure to process and extract information from signals. They use acoustic cues in many contexts, including predator avoidance, competition, mate attraction, courtship, and phonotaxis. And the few papers that we do have on invertebrates primarily focus on airborne sound that overlaps with human hearing. However, airborne sound is actually the least common modality for communication. And so I just want to show an example here of basically the hearing range of a cricket. And so it's very broad. That you see here, the most sensitive is about 5 kilohertz, which overlaps with the conspecific calls, but it goes all the way up into the ultrasonic range as well, and that's for predator detection. And they're also sensitive to substrate-borne vibrations. And so that's important when we're studying noise, because anthropogenic noise, it has multiple components, both airborne and substrate-borne, both far field and near field. So we have all these components, and you know, invertebrates can detect all those components. Most of them can. But for my talk, I'll actually be talking about noise in the far field, okay? So let me talk about kind of some of those impacts of noise on invertebrates. So the first, we have airborne noise. Again, I've broken up airborne noise into two different parts. So we have sonic, which is 20 kilohertz or 20 hertz to 20 kilohertz, which overlaps with the range of human hearing. And then we have ultrasonic, which is over 20 kilohertz outside the range of human hearing. And the main sources of airborne noise are traffic, energy development, military activity, and urban activity. So the maps here on the side show both kind of our traffic corridors in the United States. So the top one is air traffic over the United States on any given day. And then the bottom one shows the roads. You're never more than about 100 miles from a road in the United States. So that's a lot of noise that these animals are subject to just from traffic alone. And so invertebrates rely on airborne sounds for many different functions. So one is for species identity, localization of potential mates, predators, or rivals, mate quality, courtship coordination. They also use airborne sound to detect or deter predators, and that's typically in the ultrasonic range with bats. However, with ultrasound, it's largely understudied as a potential noise source. So we really don't have a lot of information about ultrasound as a noise source. So what do we know about how noise actually affects invertebrates? So we've seen changes in signals. So cicadas and grasshoppers have been shown to adjust their frequency. So grasshoppers at roadside areas call that a higher frequency, but we also see some temporal changes too. Crickets decrease the time tree crickets decrease the time they spent calling. And now these shifts in signal could potentially lead to changes in female preference. And frequency is really important for invertebrates. Being so small, it's actually considered an honest signal for body size in assessing the quality of a potential mate. We also see changes in phonotaxis. Female crickets have been shown to have trouble locating males, which could potentially decrease mating opportunities. Then we have signal interference. Ormia parasitoid flies rely on airborne signals to locate potential hosts. And when that sounds are masked, it decreases their ability to locate hosts. We also see an alteration to community structure. So when there's shifts in the presence of some species or shifts in their behavior, that can shift the overall community structure. It can also change predator-prey dynamics. So if predator cues are masked, then you might see an increase in predation. However, you also might see when predators avoid noisy areas that they could use these noisy areas as a predator shield. Again, we just need more research on that. So although there's not a lot of papers, there are hypotheses about how invertebrates can basically avoid the impacts of noise. So the first one is they can just avoid noisy areas altogether. We can also maybe temporal shifts. However, with these temporal shifts, it may be a little harder for invertebrates because many insects call in mixed species aggregations, and they're already temporally partitioning their signals to not overlap with other similar species or other conspecifics. So that would add a lot more pressure. And also, they typically call at dawn and dusk, which is also happens to correlate with the rush hour traffic. And we probably won't see really a shift away from that dawn and dusk calling just because of the optimal atmospheric conditions for these animals. So again, temporal shifts, will we see those? We're not really sure. We see an increase in amplitude for vertebrates. So far, nothing has been documented about shifts occurring within vertebrates. And then we could potentially see frequency shifts. However, the plasticity in the response to noise is actually limited due to their body size and to their hardened exoskeleton. So we may not see as much behavioral plasticity that we see in vertebrates like birds, but we may see the developmental plasticity. So the grasshoppers that shifted their frequency at roadside noise, when those nymphs that were collected at the roadside were taken into a quiet environment and reared, they still produce songs at a higher frequency. So we may be seeing a developmental plasticity on that side for frequency shifts. Now, looking at on the receiver side, the neurofilters may help to increase the frequency of conspecific calls. And we already see this on crickets that live in noisy areas compared to crickets that live in quieter areas. So this black line here is a tropical cricket. And so there's already high levels of biotic background noise. And as you can see with the hearing sensitivity, it's very much more narrow. Okay, so they don't hear as much into the higher ranges. And then for the crickets, these two crickets here that live in a field in Europe, they have much broader frequent hearing at those higher frequency ranges. So this is kind of an example, you know, of we may see shifts neurologically as well. Okay. So next we have substrate-borne noise that's again is a potential source. And 90% of all animals use substrate-borne sound, okay? And typically those are frequencies less than 1,000 hertz. And airport traffic, construction, railroads are the main sources of noise. And road noise that's less than 2,000 hertz also has the potential to add to the substrate-borne noise. Now with substrate-borne noise, because it doesn't attenuate as fast, it has the potential to be detected at longer distances from the source, okay? However, it is largely left out of studies of anthropogenic noise. Now, because of that, when we're looking at the impacts of substrate-borne noise on invertebrates, we're not investigating it in the context of anthropogenic noise. It's just investigated in general of just substrate-borne noise, okay? So this is basically what we've seen so far for the response to just general substrate-borne noise. We still see changes in signals. Female stink bugs adjust the frequency, but not the temporal components. And male stink bugs actually respond less frequently to female signals. And wolf spiders are less likely to mate in the presence of white noise. And then with male face leafhoppers, they basically cease calling for that first part of their call. And so because we know anthropogenic has these broad components to it, or it's multimodal, we need to really kind of increase that studies. And so that kind of brings me to my first question of my dissertation. How does interference by airborne sonic and ultrasonic and substrate-borne vibrational noise impact a female's ability to successfully locate signaling males? And for the study, I use Australian black field crickets with Teleogryllus commodus, and they rely on male acoustic signals to basically locate them. And phonotaxis is driven by the female's ability to detect a signal of interest. And noise presents a challenge to receivers. If the receiver cannot separate the signal of interest from background noise, errors are unavoidable. And noise can potentially lead to receivers missing a detection, which can result in a loss of mating opportunity, or noise can cause false detections, which can be energetically costly to the receiver. And like many other invertebrates and insects, black field crickets rely on sounds in the sonic range for advertisement and localization of conspecifics. And the ultrasonic range is for predator detection, specifically bats. And for substrate-borne noise, or substrate-borne sound, courtship and terrestrial predator detection. So here's just a little background on the Teleogryllus commodus, called the black field cricket. They're basically found throughout Australasia, live in urban areas, forests, and heath. Females can lay up to 2,000 eggs in a lifetime. They're really easy to rear in the laboratory. And because they're larger than many other crickets, their neurosystem is very well studied because it's just easier to study the neurons with them. And it's just very well known. And it's basically used as a model system throughout the grillid taxa. And this system relies on females' ability to detect and locate males, and the males typically call from their burrow. Okay, so for hearing, auditory reception. So their ear, or their tympana, is basically on their foreleg. So here you can see their tympana on their foreleg there. And for auditory reception, they use the tympanal organ. And so here they have scolopedial cells that are connected to the tympana, and that basically sends a signal to the prothoracic ganglion, okay? And those scolopedia cells, are the organization allows for frequency discrimination. And the tympanal organ is basically tuned to conspecific calls and the high frequency of the predators. Okay, and so you can kind of see that closer view of the tympana, and then this is basically where it's at. And for being so small, they're actually really accurate at locating a source, even though usually the distance between their two ears is usually smaller than the sound wave. So they really have complex hearing, and it's very very exact. Okay, so for substrate-borne vibrations, they use the subgenial organ, okay? And this is actually found in all of their legs. So they use all 6 legs to detect substrate-borne vibrations when they're in contact with the ground. And actually each leg is sensitive to different frequency ranges. And in their foreleg, so here's some examples of the Scolopendra in the subgenial organ, and that's just free floating. So it's not connected to any structure like the Scolopendra and the tympanal organ are. That's just kind of floating there. And it's displaced with the vibrations. Okay, and so you can see in the foreleg here that says subgenial organ and the tympanal organ are very close together. And they basically integrate into the prothoracic ganglion in the thorax. Okay, and so the subgenial organ is actually well developed by the first instar and fully developed by their fourth. Now, this is different from the tympanal organ that starts to develop in their fourth instar and isn't fully developed until their final instar. And the ear, or the tympanum on their foreleg, is not fully exposed until that final instar. Okay, so that's kind of some of the background of the hearing capabilities or the sensory system of the black-field cricket. Okay, so now I'll get into my methods for how I did that, my experiments. So I created 3 different noise sources. So the first one was substrate-borne vibrations, and that was from 100 to 1,000 hertz. I had 3 intensities, and because it's substrate-borne vibrations, it's measured in velocity. So I had 3 intensities at 1 millimeter per second, 5 millimeter per second, and 15 millimeters per second. Ultrasound ranged from 20 kilohertz to 45 kilohertz. And that overlaps with the hearing sensitivity of their close relative, I guess, of the Teleogryllus oceanicus. So that was the most sensitive hearing range in the ultrasonic range for those species. So I adapted it to Teleogryllus commodus. And also the intensities were at 70 decibels, 75 decibels, and 80 decibels. Anything lower than 60 decibels, they can't really detect in the ultrasonic range. Okay, and then for the sonic noise, which is 20 hertz to 20 kilohertz, my intensities were 67 decibels, 70 decibels, 73, 76, and 79 decibels. So in a semi-anechoic chamber, I ran single speaker trials using virgin females, so they were more receptive to male calls. Male calls were broadcast at 73 decibels from a single speaker on the side of the arena, and it was randomized by location. So here's the speaker that I would broadcast the male call from. Substrate-borne vibrations were produced by a transducer located beneath the arena. So there's just in contact with the arena underneath. And airborne sonic and ultrasonic noise was broadcast from speakers located above the arena. And all sounds were calibrated at the center of the arena right here. And that was the starting point of the female. Okay. And 50 females were used for each noise treatment, and females were exposed to each intensity of the noise treatment she was used in. And then one control that involved just the male call and no noise. Okay. And so females were placed in the center of the arena. The male call and noise were broadcast simultaneously, and her behavior was recorded using a webcam suspended above the arena. So I'm going to play 2 videos of the trial so you can kind of see some of her behaviors and basically what I was analyzing. Okay, so this is a control for the ultrasonic treatments. And so here you can hear the male call coming from the right speaker, but no noise is being broadcast right now. It's just the control to make sure she is actually receptive to that male call. And so once the female locates the speaker, the trial's over. Okay, so for the next one, it's the same female, but with ultrasonic noise being broadcast from above at 80 decibels. So for this one, the male call is being broadcast from the left speaker. You can kind of hear the clicks of the ultrasound noise. So for this female, it takes her a little longer to locate the speaker. a little slower. Okay, but she does eventually find that speaker, and it does take her a little longer than the control. So once I have those videos, I analyze them in a program called Ethovision. And so here are two pictures of tracks that I analyzed. So the first one shows a control trial, and it shows the track that the female traveled to get to the speaker. And that is a control for the airborne sonic noise. And over here is an example of the same female with the airborne sonic noise at 73 decibels. And so once I had those tracks, basically Ethovision analyzed them for multiple variables. And those variables included distance, velocity, duration, both latency to start. And so that means of once she left this circle here, that was latency to start. And then the total overall duration, meander, both total and mean, and both acceleration, minimum and maximum. So once I had that data, I was able to do a repeated measures Friedman's test. And so here are my results for that. So first is for substrate-borne vibrations. So looking at latency, it shows that, oh, and remember that substrate-borne sound is used for courtship and to detect terrestrial predators. So what I found was that at the highest intensity, latency decreased. So she was quicker to move outside that circle in the presence of noise at 15 millimeters per second. Okay. And also for duration, I saw an increase in the amount of time it took her to locate the speaker at the 1, 5, and 15 millimeter per second treatments. And then for distance, so in that presence of noise, didn't matter what the intensity was, the distance the female traveled to locate that speaker also increased. And then for velocity, noise increased the female's velocity, okay? So as the female was searching for the speaker, the velocity increased. And so those were the variables that we saw a significant impact in. And so in the presence of substrate vibrations, latency decreased. So the female was quicker to move from the center as the noise increased, and duration also increased. So she spent more time locating the male. She also traveled a greater distance to locate the male in the presence of noise, and she traveled at a faster velocity. Okay, so for ultrasound noise, so this was also, so females use, or crickets use ultrasound to detect predators. And so what we saw with that is in latency, at the highest intensities, latency decreased. So again, she was quicker to move from the center at 75 decibels and 80 decibels. Here again, we see an increase in duration. So again, she's spending more time locating the male regardless of intensity of sound. So just the presence of sound increased her duration. Same with distance. So she also increased the distance traveled to locate the speaker regardless of the noise intensity, as well as velocity. So the velocity increase for the highest two intensities at 75 decibels and 80 decibels. So she traveled at a higher velocity for those two intensities. And also for max acceleration at the highest two intensities, her max acceleration increased. So as noise intensity increased, so she was faster to move from the center of the arena, she spent longer locating the males, traveled a greater distance at a faster velocity, and her max acceleration was significantly higher at the highest intensity of noise. So this last one is for the sonic noise. And so remember, they use sonic noise to locate and to assess the quality of potential mates. And so for this one, again, we're seeing an increase in duration regardless of intensity. So again, just the presence of noise is causing her to increase the duration. And we see another increase in distance, so traveling greater distance to locate the male. And then also an increase in velocity. And so for sonic noise, those were the only three variables we saw increased. It was duration, distance, and velocity. And so overall, looking at these behavioral shifts, they potentially could have consequences. So in energy expenditure, when they're increasing their velocity or the distance traveled, you know, you might see more energy expenditure. But then when we're seeing an increase in duration, that could also expose them to more predation. So these shifts may have potential consequences. So in conclusion, we're seeing that different forms of noise do impact the female's performance in locating a male. Duration, distance, and velocity increased in all treatments. And for substrate vibrations and ultrasound, latency decreased with an increase in noise intensity. So females were quicker to move from the center of the arena with substrate-borne noise and ultrasound. And noise is present in multiple modalities, and anything outside the range of human sensory system is often overlooked. And so we really need to think more broadly about how we investigate anthropogenic noise. And so we also need to take into consideration the sensory system sensitivity of the animal we are working on. You know, these animals have broad ranges of hearing, but some animals may be more specific for their sensory system. So we really need to take that into account. And beyond research on noise, for those of us doing behavioral studies and keeping animals in the lab, we need to be aware of sources of noise that may be impacting the animals we're working with. You know, HVAC systems often produce ultrasonic noise. And so how does that affect the animals that we're keeping in our labs and their behavior and potential physiology? And we really need to expand our research to include that broad range of noise that's potentially affecting animals in the field and in the lab. So for future directions, I'm actually working on right now doing more trials that I'll focus on female choice. So it'll be similar to those arena trials I showed you guys that I was doing with this phonotaxis study, but I'm going to be focusing on choice tests. So how does the female make high stakes decisions in the presence of noise? And we'll also be looking at the neurological response of crickets to noise as well. But overall, we need more field studies to quantify all the components of anthropogenic noise, not just the noise in the human sensory range. And we also need to expand the taxa represented in studying the impact of noise on animals. Okay, thank you. Any questions?
Thank you, Jesse. You've got a round of applause from our room. And people are chatting in and iconing in their applause from virtual. I am going to open up the chat right now. I think you can still stay shared.
Okay.
I just wanted to say thank you for presenting on this topic. I know that this is terrestrial, but from the underwater world, seeing multimodal studies is really nice because, as you noticed, in the terrestrial world with the substrate-borne studies, the same is true for particle motion in the underwater world, underrepresented compared to the sonic and ultrasonic air pressure, or water pressure. So I just wanted to thank you for really bringing up the topic of multimodal acoustic communication. We don't hear that very often.
Yeah, and this is just the far field. We don't even talk about near field, you know, noise as far as it impacts, and most invertebrates are very sensitive to particle movement. So near field noise is also something else we need to expand into as well.
Michelle Halverson, sitting in our room here, just echoed your invertebrate comment. Sure, questions, please.
Okay, go ahead.
Hi, Jesse. Great talk. I love this.
Thank you.
But I'm ignorant on most of it. So I'm going to ask a silly question that you may have explained and I fell asleep. You kept talking about the female in the singular. How many females did you have?
Oh, sorry. I did 50 for each treatment.
50 different females.
Yes. And they were exposed to every intensity plus the control. So it was, yeah, 50.
Okay.
I think I had to analyze like 300 videos.
Okay, so my questions now go into two different paths. If it had
Okay.
been with a small number of females, I was going to ask if they could learn to deal with the noise because you had a lot of overlap in your baseline compared to your noise level experiments. But I'm going to put that one aside. My other one, so knowing now that you had a large number of females, from your error bars and the overlap in your results, it seems like some females were actually pretty good at dealing with noise. Overall, maybe the trend was in the direction you said, but it seems to be pretty mushy. Like some can actually, it doesn't bother them, or at least some of the experiments.
Yeah, and well, when you think about it, like with ultrasound and substrate-borne vibrations, there's no masking going on because typically masking is the big issue, right? With detecting and finding and locating the male. So for substrate-borne vibrations, ultrasound, it may just be that there's more information to process, so it may take longer. So it's not stopping them from locating the male. It just may take longer because they need more time to process. And also those two noise treatments also signify, at least for them, potential predators. That's what they use ultrasound and substrate-borne. So again, for those, the shift in time may be telling that they're just processing it or just, is this a predator? Is this not? So I don't think ultrasound and substrate-borne vibrations are hindering or are preventing them from locating the male. It's just kind of affecting the path to the male. It's the airborne sonic noise that it may be more of an issue with masking and not being able to locate the male.
Gotcha.
I'm going to ask a chat question here. For the p-values, what statistical tests were used?
I used the Friedman's test because it was a non-parametric Friedman's repeated measures test.
I had a question about your noise recordings. Were they simulated or did you record them from nature somewhere?
No, these ones were simulated. So I created them in Adobe Audition.
Excellent. If you had taken them from the field, I was going to ask about pseudo replication, but seeing they were simulated, I didn't have to.
I mean, granted, like I said, we do need more out in the field, but to get the baseline of what's actually happening, this is easier to control to make sure it does overlap with the sensory system of these crickets.
Do you have any plans to use noise sources from nature or anthropogenic noise sources the crickets would encounter in their natural life?
Not for my current dissertation. I originally was going to use train noise from Kingman Farm to basically look at the both airborne and substrate-borne. I was going to play back both multimodal, but because of the drought last year, I couldn't find any crickets. So that had been the plan, and I would really like somebody maybe to take over and do that later. But unfortunately, with environmental conditions being so variable with climate change, it was hard.
I'm hearing postdoc project.
Yeah, I was really excited about that because I recorded both the airborne and substrate-borne as the train went by at Kingman Farm, and I set it up to play it back. And I was going to look at the response of males, singing males to it, and I just couldn't find males for one season. They were completely gone.
Didn't have that problem this year, I'll say that much with all the rain.
Yeah.
Any other questions, either from the room or from online?
Oh, he has another.
Ian has a question here. Can you project loud enough?
Sure. Can you hear me okay?
Yeah.
All right. Yeah, just I guess I should introduce myself. I'm Ian. I'm a new postdoc with Jen. And yeah, my PhD was also with invertebrates, squid responses to noise. So I always appreciate learning more about people who work with invertebrates. And I want to also echo what Jen said about the multiple sensory modalities doing airborne and substrate-borne. I thought that was great to see. But my actual question was, I was curious if anything had been studied with these crickets, or if you plan to look at things like hearing threshold shifts and how that might play into kind of the changes in behaviors you're seeing.
Yeah, so actually my third chapter is going to be looking at the thresholds of their neurons. So I'll look at the thresholds and then how those thresholds are impacted by noise. So we will be getting to those neurological thresholds in 2 of my chapters, actually. Haven't done those yet, but I'm gonna get to them.
Cool, thank you. I look forward to hearing about that.
Yeah.
And another question from the room?
So it's Tony again. I'm still representing the ignorance portion of the acoustics who've never done these types of experiments, but how does one exactly get a hearing sensitivity curve on a cricket?
So originally what they used to do is they used to tether them and have them fly. And as they adjusted their behavior, they would determine that they could hear sound. Now, luckily, we can do neurological responses. So basically what I can do is I expose their neurons and I can basically use a hook, probe them, and then we get the action response of the neurons as we expose them to noise. And because these are invertebrates, we can keep them alive while we do this. And so we can get the neuro response to noise, which is obviously a little better, I guess, than the behavioral response, because it's not so subjective, I guess. So yeah, we basically look at the response of the neurons, the action potentials there when we play noise to them.
Right, I understand that. So I see there's another chat, but I'm going to keep you here for one more second. So you would have to do that for your study for the 2 different mechanisms, the 2 different hearing physical mechanisms for hearing that you used.
Yep.
For the substrate and the sonic. Okay.
Yeah, and we'll do both sonic, so it'll be 3 treatments again. So it'll be in the airborne, it'll be sonic and ultrasonic, and then substrateborne. So I'm going to try to do all of them, because most of the hearing audiograms we have for crickets and a lot of other insects are actually airborne sound. And we have a few that looks at the sensitivity to substrateborne vibrations, but not a lot. It's mainly their sensitivity to airborne.
Super cool. Maybe I'll come to a postdoc with you. This sounds fun.
Yeah, there's a question in the chat. Do you think using wild crickets who may have been exposed to anthropogenic noise in their environment impact the results in any way, opposed to using crickets naive to those noises with naive to those noises with no previous exposure?
It could potentially, and that was actually a question I had an undergrad in here ask, because I didn't have enough time to ask that question. But we did take crickets from noisy areas next to railroad tracks and roads and kind of did a similar trial to crickets in quieter areas. And so we're still working on that to see if there is a difference. Now that one unfortunately has a really small sample size, but it is something we need to think about because, like I said, when we see those grasshoppers that changed their frequency, that seemed to be developmental plasticity. So I think it is important for us to look at these wild types and see, is there a difference between ones that, do live in noisy corridors and ones that live outside? And that could potentially be developmental plasticity. And I would actually be really interested in looking at the neurological, like doing an audiogram comparing 2 different, crickets at quiet sites and crickets at noisy sites, and just seeing if their hearing capabilities have changed at all, but yeah, so these are all questions that because there's not a lot of work in it, there's so much research left to do in this, and they're really easy to work with, they're easy to rear in captivity. You also don't have to go through IACUC,
Thanks. At last chance for questions here. I think, Jesse, this is a really great trial run for your thesis defense, because all of these are great questions and you've done a really good job answering them.
Thank you.
Last call, going once, going twice. Okay, final round of applause for Jesse. Nice job. And hopefully we'll see everybody back here next month for our next acoustic seminar. Thanks so much, everybody, for
thank you
for tuning in. I saw people from other universities, from industry, from so that's super great to see such a diverse audience here. And I know in some places there's more than one person like ours in a room. So Thank you, everybody, for your time, and hope to see you next month. Stop recording.